eukaryotic cells have an endoplasmic reticulum (ER). The existence of ER was discovered in 1945 by Porter, Plaude and Fulham. The base structure and composition of the ER is similar to the plasma membrane, though it is an extension of the nuclear membrane. The ER membrane typically constitutes more than half of the total membrane of an average animal cell. Prokaryotic organisms do not have organelles and thus do not have an ER. There are two distinct types of ER that perform different functions within the cell: the rough ER and the smooth ER. Parts of the ER are covered with ribosomes, and their rough appearance under electron microscopy has led scientists to call them as rough ER or ER. Other parts of the ER, which are free of ribosomes and smooth in appearance, are called as smooth ER or SER. Cells that synthesize steroid hormones from cholesterol have an expanded smooth ER compartment to accommodate the enzymes needed to make cholesterol and to modify it to form the hormones. Smooth endoplasmic reticulum consists of tubules and vesicles that branch, forming a network. In some cells, there are dilated areas like the sacs of rough endoplasmic reticulum. The network of smooth endoplasmic reticulum allows increased surface area for the action or storage of key enzymes and the products of these enzymes. In the case of smooth endoplasmic reticulum in muscle cells, the vesicles and tubules serve as a store of calcium, which is released as one step in the contraction process. Calcium pumps serve to move the calcium. In adrenal cortical cells as well as steroid producing cells in the gonads, the smooth endoplasmic reticulum serves to metabolize the steroids and produce the final steroid hormones. After the side chain of cholesterol is cleaved in the mitochondria, the product is passed to the smooth endoplasmic reticulum and further modified. Smooth ER is abundant in cell types that are particularly active in lipid metabolism. In addition, smooth ER is abundant in the liver, where it contains enzymes that metabolize various lipid soluble compounds. These detoxifying enzymes inactivate a number of potentially harmful drugs, for example, phenobarbital, by converting them to water soluble compounds that can be eliminated from the body in the urine. In the great majority of cells, area covered by RER is scantily and are often partly smooth and partly rough. They are sometimes called transitional ER because they contain ER exit sites from which transport vesicles carrying newly synthesized proteins and lipid bud off for transport as endosomes to the Golgi apparatus. In certain specialized cells, however, the smooth ER is abundant and has additional functions. The main cell type in the liver, the hepatocyte, is an example of a cell with an abundant smooth ER. It is the principal site of production of lipoprotein particles, which carry lipids via the bloodstream to other parts of the body. The membranes of eukaryotic cells are composed of three main types of lipids: phospholipids, glycolipids and cholesterol. Most of the phospholipids, which are the basic structural components of the membrane, are derived from glycerol. They are synthesized on the cytosolic side of the ER membrane from water soluble cytosolic precursors. Fatty acids are first transported from coenzyme A carriers to glycerol 3 phosphate by a membrane bound enzyme, and the resulting phospholipid phosphatidic acid is inserted into the membrane. Enzymes on the cytosolic phase of the ER membrane then catalyze the addition of different polar head groups, resulting in formation of phosphatidylcholine, phosphatidylserine, phosphatidylethanolamine or phosphatidylinositol. Assembly of membrane lipid bilayers in the ER. The ER synthesizes all of the major classes of lipids, including both phospholipids and cholesterol required for the production of new cell membranes. The major phospholipid made by RER is phosphatidylcholine, also called as lecithin, which can be formed in 3 steps from choline, 2 fatty acids and glycerol phosphate. Each step is catalyzed by enzymes in the ER membrane that have their active sites facing the cytosol, where all of the required metabolites are found. Thus, phospholipid synthesis occurs exclusively in the cytosolic leaflet of the ER membrane. In the first step, acyl transferases successively add 2 fatty acids to glycerol phosphate to produce phosphatidic acid, a compound sufficiently water insoluble to remain in the lipid bilayer after it has been synthesized. It is this step that enlarges the lipid bilayer. The later steps determine the head group of a newly formed lipid molecule and therefore, the chemical nature of the lipid bilayer, but they do not result in net membrane growth. The 2 other major membrane phospholipids, phosphatidylethanolamine and phosphatidylserine, as well as the minor phospholipid phosphatidylinositol (PI), are all synthesized in this way. The synthesis of these phospholipids and translocation on the cytosolic side of the ER membrane allows the hydrophobic fatty acid chains to remain buried in the membrane, while membrane bound enzymes catalyze their reactions with water soluble precursors, for example, CDP choline in the cytosol. Because of this topography, however, new phospholipids are added only to the cytosolic half of the ER membrane. To maintain a stable membrane, some of these newly synthesized phospholipids must therefore be transferred to the other, that is luminal half of the ER bilayer. This transfer does not occur spontaneously because it requires the passage of a polar head group through the membrane. Instead, membrane proteins called flippase catalyze the rapid translocation of phospholipids across the ER membrane, resulting in even growth of both halves of the bilayer. In addition to its role in synthesis of the glycerol phospholipids, the RER also serves as the major site of synthesis of 2 other membrane lipids, cholesterol and ceramide. The plasma membrane and the membranes of the Golgi complex, lysosomes and endosomes all form part of a membrane system that communicates with the ER by means of transport vesicles that transport both proteins and lipids. Mitochondria, plastids and possibly peroxisomes do not belong to this system, and they therefore require different mechanisms for the import of proteins and lipids for growth. It is now well understood that most of the proteins in these organelles are imported from the cytosol. Although mitochondria modify some of the lipids they import, they do not synthesize lipids from scratch. Instead, their lipids have to be imported from the ER either directly or indirectly by way of other cell membranes. In other case, special mechanisms are required for the transfer. It might be that mitochondria and peroxisomes are the only lipid poor organelles in the cytosol, and that such an exchange process is sufficient. In electron micrograph, mitochondria are often seen in close juxtaposition to ER membranes, and there may be specific mechanisms of lipid transfer that operate at such regions of proximity. Liver is the main organ in animals where most drug detoxification reactions takes place. Smooth ER is abundantly present in the hepatocyte, the main cell type in the liver. The enzymes that synthesizes the lipid components of lipoproteins are located in the membrane of the smooth ER, which also contains enzymes that catalyze a series of reactions to detoxify both lipid soluble drugs and various harmful compounds produced by metabolism. The most extensively studied of these detoxification reactions are carried out by the cytochrome P450 family of enzymes, which catalyze a series of reactions in which water insoluble drugs or metabolites that would otherwise accumulate to toxic levels in the cell membranes are rendered sufficiently water soluble to leave the cell and be excreted in the urine. Because the rough ER alone cannot house enough of this and other necessary enzymes, a major portion of the membrane in a hepatocyte normally consists of smooth ER. When large quantities of certain compounds such as the drug phenobarbital enter the circulation, detoxification enzymes are synthesized in the liver in unusually large amounts, and the SER doubles in surface area within a few days. Once the drug has disappeared, the excess SER membrane is specifically and rapidly removed by a lysosome dependent process called autophagocytosis. Smooth endoplasmic reticulum is usually the major calcium storage organelle in eukaryotic cells. However, recent findings suggest that in addition to SER, mitochondria are another important player in eukaryotic cellular calcium economy. Smooth ER in most eukaryotic cells sequester calcium from the cytosol. the release of calcium into the cytosol from the ER and its subsequent reuptake is involved in many rapid responses to extracellular signals. In plant cells, calcium is stored in vacuoles. The storage of calcium in the ER lumen is facilitated by the high concentrations of calcium binding proteins there. In some cell types and perhaps in most specific regions of the ER are specialized for calcium storage. Muscle cells, for example, have an abundant specialized smooth ER called the sarcoplasmic reticulum, which sequesters calcium from the cytosol by means of a calcium ATPase that pumps in calcium into its lumen. The release and reuptake of calcium by the sarcoplasmic reticulum trigger the contraction and relaxation respectively of the myofibrils during each round of muscle contraction. Export of proteins and lipids from the ER. Both proteins and lipids travel along the secretory pathway in transport vesicles which bud from the membrane of one organelle and then fuse with the membrane of another. Thus, molecules are exported from the ER in vesicles that bud from ER and carry their cargo first to the ER Golgi intermediate compartment and then to the Golgi apparatus. Subsequent steps in the secretory pathway involve vesicular transport between different compartments of the Golgi and from the Golgi to lysosomes or the plasma membrane. In each case, proteins within the lumen of one organelle are packaged into the budding transport vesicle and then released into the lumen of the recipient organelle following vesicle fusion. Release of hormones from the endocrine glands such as pancreas, thyroid, pituitary, hypothalamus, adrenal gland takes place by this mechanism only. The hormones are released in blood stream in the form of granules packed in ER membrane, then transported to Golgi and finally fuses with plasma membrane. ER of membrane proteins and lipids are transported similarly, and it is noteworthy that their topological orientation is maintained as they travel from one membrane enclosed organelle to another. For example, the domains of a protein exposed on the cytosolic side of the ER membrane will also be exposed on the cytosolic side of the Golgi and plasma membranes, whereas protein domains exposed on the luminal side of the ER membrane will be exposed on the luminal side of the Golgi and on the exterior of the cell. While most proteins travel from the ER to the Golgi, some proteins must be retained within the ER rather than proceeding along the secretory pathway. To study the functions and biochemistry of the ER, it is necessary to isolate the ER membrane. This seems like a tedious task because the ER is intricately interleaved with other components of the cytosol. Fortunately, when tissues or cells are disrupted by homogenization, the ER breaks into fragments and reseals into many small, around 100 to 200 nanometers in diameter, closed vesicles called microsomes, which are relatively easy to purify. Microsomes derived from rough ER are studded with ribosomes and are called rough microsomes. The ribosomes are always found on the outside surface, so the interior of the microsome is biochemically equivalent to the luminal space of the ER. Because they can be readily purified in functional form, rough microsomes are especially useful for the studying the many processes performed by the rough ER. To the biochemist, they represent small authentic versions of the rough ER, still capable of protein synthesis, protein glycosylation, calcium uptake and lipid synthesis. Many vesicles of a size similar to that of rough microsomes, but lacking attached ribosomes are also found in these homogenates. Such smooth microsomes are derived in part from smooth portions of the ER and in part from vesiculated fragments of plasma membrane, Golgi apparatus, endosomes and mitochondria. Thus, whereas rough microsomes are derived from rough portions of ER, the origins of smooth microsomes cannot be as easily assigned. The microsomes of the liver are an exception. Because of the unusually large quantities of smooth ER in hepatocytes, most of the smooth microsomes in liver homogenates are derived from smooth ER. The ribosomes attached to rough microsomes make them more dense than smooth microsomes. As a result, the rough and smooth microsomes can be separated from each other by equilibrium centrifugation. The rough microsomes, however, contain more than 20 proteins that are not present in smooth microsomes, showing that some separation mechanism must exist for a subset of ER membrane proteins. Some of the proteins in this subset help to bind ribosomes to the rough ER, while others presumably produce the flattened shape of this part of ER. Smooth ER is more tubular than rough ER and forms a separate sealed interconnecting network. The tubules and sacs are all thought to interconnect so that the ER membrane forms a continuous sheet enclosing a single internal space. Smooth ER is devoted almost exclusively to the manufacture of lipids and in some cases to the metabolism of them and associated products. In liver cells, smooth ER enables glycogen that is stored as granules on the external surface of smooth ER to be broken down to glucose. Smooth ER is also involved in the production of steroid hormones in the adrenal cortex and endocrine glands. Depending on cellular functions, the smooth ER can double its surface area within a few days, returning to its normal size when assault has subsided. The contraction of muscle cells is triggered by the orderly release of calcium ions from the smooth endoplasmic reticulum.